So the first one is from Dr. Ryan Potts at St. Jude's, and this is actually a second year of funding for Dr. Potts, who is an expert in understanding how the MAGEL2 gene or the MAGEL2 protein functions within the cell. And he's found that MAGEL2 is important for recycling within the cell. So we know that the cell reuses a lot of the cellular receptors that sit out on the surface, and there's a pretty specific process by which those receptors kind of get recycled. And he's found that the MAGEL2 protein is important in that normal recycling process. And working with Dr. Larry Reiter, who FPWR has also funded, and using the dental pulp stem cell, so hopefully many of you have sent in your baby teeth to Dr. Reiter, who then grows neurons and gives them to Dr. Potts to study this phenomenon. Dr. Potts has shown that in PWS and also in Chaff-Yang syndrome that this processing, this recycling, isn't working correctly. So he's using stem cells derived from the baby teeth to study how the stem cells are developing into neuronal cells and how the recycling goes wrong in these cells. So their team is, in the second year, is going to be building on their experience so far and examining some variants of the MAGEL2 gene to see if they can recover the function that is lost when MAGEL2 is lost. So we're excited about this study because Dr. Potts is really an expert in the role of MAGEL2 in endosomal recycling. This recycling defect has, it really could explain many of the cellular defects in PWS if those receptors are not getting back to the cell surface the way they're supposed to. And this pathway can also serve as an excellent target for drug development. So your next question, or Dr. Potts' next question, will be, can we use drugs to recover that recycling ability? So the ultimate goal of this project is to understand the role of MAGEL2 in this recycling and how loss of MAGEL2 disrupts that process, and ultimately whether drugs can compensate for that.